Sandwich ELISA Operation Guide. Before the experiment, please verify the contents of the kit and the instruction manual. If you have any questions, contact us if needed. Preparation of standard working solution and sample. Centrifuge sample for 5 minutes at 1000 G, 4 degrees Celsius. Centrifuge reference standard for 1 minute at 10,000 G, 4 degrees Celsius. Add 1 milliliter of reference standard and sample diluent. Stand for 1 to 2 minutes. Take 7 EP tubes. Add same volume of reference standard and sample diluent to each tube. After standing, mix the standard thoroughly with a vortex meter of low speed. Centrifuge the solution for 1 minute at 800 G. Take the same volume of standard solution and dilute it. Mix thoroughly with a vortex meter. Dilute the standard in turn. The last tube is regarded as a blank. Dismounting of micro ELISA plate. Loosen the strips and push the bottom. Remove the strips and place them in a spare aluminum foil bag or on your own plate frame. Prepare wells according to experiment requirements. Pipetting. Add 100 microliters standards from low to high concentration. Avoid touching the inside wall. Vertically add 100 microliters prepared samples. Avoid touching the inside wall. Cover the plate with sealer and mark the name, time and abbreviation. Preheat the incubator in advance. Pay attention to the temperature. Incubate at 37 degrees Celsius for 90 minutes. Preparation of biotinylated detection AB working solution. Take out the appropriate volume of biotinylated detection antibody diluent. Add the concentrated biotinylated detection AB to the diluent to prepare 100-fold diluted working solution. Invert it gently for 20 times and set aside. Take out the plate. Avoid touching the bottom of the wells. Decant the solution from each well and pat it dry with clean absorbent paper. Pour the solution into the loading tray. Vertically add 100 microliters to each well. Cover the plate with sealer and mark the name, time and abbreviation. Incubate at 37 degrees Celsius for 60 minutes. Preparation of 1 times wash buffer. Take out the appropriate volume of double distilled water. Add the concentrated wash buffer to the double distilled water to prepare a 25-fold diluted working solution. Mix it with the magnetic stirrer for 5 to 10 minutes. Preparation of the HRP conjugate working solution. Take out the appropriate volume of HRP conjugate diluent. Add the concentrated HRP conjugate to the diluent to prepare 100-fold diluted working solution. Invert it gently for 20 times and set aside. Take out the plate. Avoid touching the bottom of the wells. Tear off the plate sealer gently to avoid spillage. Set parameters. Wash. Pat it dry and replace the absorbent paper. Pour the solution into the loading tray. Vertically add 100 microliters to each well. Cover the plate with sealer and mark the name, time and abbreviation. Incubate at 37 degrees Celsius for 30 minutes. Color development. Take out the plate. Avoid touching the bottom of the wells. Tear off the plate sealer gently to avoid spillage. Set parameters. Wash. Pat it dry and replace the absorbent paper. Pour the solution into the loading tray. Vertically add 90 microliters to each well. Cover the plate with sealer and mark the name, time and abbreviation. Incubate at 37 degrees Celsius for about 15 minutes. Termination reaction. Take out the plate. Avoid touching the bottom of the wells. The first 4 wells will show a clear blue gradient after color development. Tear off the plate sealer gently to avoid spillage. Vertically add 50 microliters to each well. The color turns yellow from blue when the stop solution is added. Data processing. Gently wipe the bottom of the plate. Put the plate into the microplate reader and determine the optical density. Plate layout. Data layout. Enter the plate. Shaking type, continuous. Shaking speed, medium. Duration, 5 seconds. Absorbance, filter, 450 nanometers. Measurement mode, normal. Start. Save. Measurement. View data. Save the data.